Welcome to Data in Biotech, a podcast from CorrDyn where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we're excited to be joined by Markus Gershater, Chief Scientific Officer and co-founder of Synthace, a digital experiment platform built for high-performance life science R&D teams that help them run more powerful experiments and accelerate scientific progress. During this interview, our host, Ross Katz, speaks with Markus on what's broken about how laboratories across the globe run biological experiments at scale, the opportunity that exists for researchers in striving to achieve experimental design automation processes, why as an industry we must move towards implementing infrastructure that enables multifactorial experiments versus one-factor experiments, and the role of AI in making sense of complex systems. Here we go.
Markus Gershater, welcome to the Data in Biotech podcast.
Thank you very much. Really glad to be here.
Appreciate you joining us. So just to get us started, in a minute or two, could you just give us your background and an overview of your career to date?
Sure. Yeah, I've had a pretty varied career. The thing that ties it all together is essentially biology, a passion for biology. So I actually started out in plant biochemistry, although to be honest, it could have been any area of biology. Like pretty much every stage of my career, I've been quite opportunistic. And in this case, it was an opportunity to work at Kew Gardens for a year here in London, which is absolutely incredible botanical gardens. For anybody that's not been there, I recommend it very highly. And I just jumped at it. Like since then, I've done bioprocess development, I've done synthetic biology. Or latterly, I've learned an awful lot about like how therapeutics are discovered and developed. So yeah, the other common theme is that I've always been on the interface of different disciplines, those interfaces with biology, right? Chemistry, maths, computer science. And I think it's always that interface where the most interesting stuff tends to happen. I don't tend to be like interested as much in like where things get like really pure and like super deep. I'm all about like big concepts coming together, big ideas, different mindsets, and what you can learn from people who've essentially trained in a very different discipline to your own.
So that multidisciplinarity and the way that the different disciplines feed into each other and get synthesized together.
And I wouldn't call myself multidisciplinary, really. I am still very much a biologist. I guess I'm part biologist, part businessman these days, as much as it almost makes me slightly weird saying it. But yeah, I guess it's that skill of being able to talk across disciplines to really try and understand what someone's saying, even though they might be saying it in a really unfamiliar way, unpacking things, that kind of thing. I think it's a lot about communication more than anything. I was chatting with someone the other day about like communication can be a real superpower, but I think it's one that's sometimes underestimated, right?
Your career's taken this interesting journey at the intersection of these different disciplines. Why have you gone on this journey? What's motivated you to take the path that you've taken?
Biology, in a word. So I've always wanted to be a biologist. I haven't ever thought of being anything else. My dad's a biochemist by original training, and so I'm sure that rubbed off on me. But it's like biology is the most incredible phenomenon you can imagine. And I think once you start learning about it, probably outside of a school environment, because frankly, the way that it's taught at school, certainly in the UK, is just dreary. Like, no offense to my biology teachers, it's more the curriculum. It was just like, it's just lists of facts to learn. But when you actually understand about these hugely complex systems and how they evolved and like what they've evolved into, then it never ceases to delight and surprise. And I'm continually delighted how wrong I was yesterday about biology. It's continually fascinating. But it's also like one of the most frustrating disciplines. Like it's carried out in a hugely manual, sort of step-by-step artisanal way. You've got the tools that most scientists are using for doing biology. It's pipettes and a copy of Excel and frankly, incredible amounts of hard work and patience and stoicism, right? And when you contrast that with that huge, unfathomable complexity and of the discipline, of the phenomenon that we're trying to study, you just take a step outside it and look at it from the outside, and it's utterly baffling. And that frustration is a motivation of sorts as well.
Right. So that frustration, I'm imagining, is part of the reason why Synthace came into being in the first place. Is that right?
Right. I think it's a combination of the enthusiasm and the frustration, right? I think I've got a completely unbounded belief almost in the good that biology could do if we could work with it as effectively as we possibly can. And so then I guess that just makes the frustration even more so, right? Because you've got all of these solutions just there. We have this absolutely incredible phenomenon that we can work with. That's digitally programmable, right? But it's something that just feels like it should be something that we should just be able to turn to any big problems that are facing humanity. And yet at the same time, anybody who's actually tried to do it knows how incredibly hard it is. I think it's quite funny sometimes, not necessarily funny, but it's an interesting phenomenon if you look at like synthetic biology startups, because often they'll start out in this very kind of starry-eyed way of, hey, we can program biology and we can solve the world's problems. And then very quickly you realize, hang on a second, this system, it doesn't lend itself to easy engineering and predictable function, right? As any bioprocess engineer will be very glad to tell you, right?
So biology is just by its nature a noisy process with lots of variables coming into it. And also the methods that people are using to experiment with that process are very manual in nature and error prone in nature, which adds additional noise to this already noisy system. And am I understanding you correctly?
Yeah, and it's not just noise. So I think there's a really fundamental feature of biology which I think is under-considered, and to try and compensate for this, I bang on about it all the time, right? And that is that biology, I think, becomes a really fascinating thing when you think about the process by which it came into being. So this process of evolution. And evolution is like a fascinating phenomenon in its own right, in that it has absolutely no regard for how complex a system is that it's making, or how incredibly intertwined and interdependent that process might be. So long as it gives some kind of fitness advantage, then evolution will select for it. And it doesn't matter how messy it is, right?
Mm-hmm.
And what you end up with then is this ridiculously interconnected filigreed system, which nonetheless has these absolutely amazing properties, right? But it's not clean, and it's in no way designed, and it's not particularly easy to design with. And so when you accept it as, okay, it's this emergent system, it's something which is many things stacked upon another and relying on each other, then you realize that it isn't just the tools that we're using that need to change, but it's also the way we're experimenting, which needs to change. And what I mean by that is we can't just be going in with classical experiments where you are fixing everything but the one factor that you're most interested in at that particular moment, because the way that factor affects your system will be dependent on how other factors change as well with that system. It's all interlinked. And we've seen over many years now, and I've seen prior to Synthace and now at Synthace, how way more powerful it is if you do properly designed multi-dimensional experiments to explore biology. So I think, yeah, we talk a lot about the tools. Yeah, it shouldn't be manual. It shouldn't be copies of Excel. It shouldn't be people just spending their incredibly educated lives, 14-hour days in the lab pipetting stuff, right? It absolutely shouldn't be that. But at the same time, we don't want to be doing dumb automation either. It needs to be stuff which is doing really elegant, structured experiments, which give us the most insight possible into this system that we're trying to learn about.
So could you just provide an overview of Synthace's platform and how it helps R&D teams working for biotechnology companies to experiment in smarter ways and to drive that broader learning that you're talking about driving?
Yeah, I've been talking about some very high level concepts and some very high aspirations, right? What does it look like when the rubber meets the road? So Synthace is a digital experiment platform, which is not a term I'd expect anybody to understand what it means, because we came up with it ourselves to try and describe what it is we do, because we don't fit into a particularly neat bucket. We get a lot of questions of, oh, so you're like an ELN? And we're like, no. So that's an electrolyte lab notebook. Or, oh, so is it like LIMS? It's like, no. Oh, your automation software. It's like, so we call it a digital experiment platform. And what that means is essentially a set of digital tools and capabilities which help the scientist through that process of running an experiment. If you look at the process of running an experiment, it's a hugely involved process of calculations, logistics, experimental design, just mapping things out about which liquids they have to put in what places, stock concentrations, booking the equipment, making sure the equipment's actually working, making sure they've got the reagents they need in the cupboard and their colleague hasn't just used it. There's just a huge amount of tedious stuff. And we look to essentially give people the digital tools that allows them to alleviate a lot of that tedium. So essentially what a scientist can do in Synthace is they can map out what they want to do in their experiment in a no code drag and drop interface, which basically lets them say, okay, I've got these samples and I want to dilute them and then I want to mix them together. And so they're thinking about what happens to their samples in their experiment as they go through it. And then from that definition, the main kind of core of the power of Synthace is our planner. So that then takes that definition and it converts it into all of the details of exactly how that experiment is going to be carried out. So this is everything from what the stock concentrations might be, how much of each liquid you need, what kind of plasticware you need, how many tips you need, and then every single liquid handling action. So every single pipetting action that's required to carry out that experiment. So then you have a fully detailed kind of map of what has to happen in that experiment to do what the scientists just defined in the system. And so then what Synthace can do is it can convert that map into automation instructions. So like we interface with the most common liquid handling automation in the lab. And so you can then send it to that automation and it'll carry out your experiment for you. And then it can gather the data that comes from the end of that experiment. But this is where like one of maybe the sort of hidden benefit comes in, right? So, you've had all of this benefit as a scientist. It's planned stuff for you. It's programmed the automation for you. You haven't had to do all the pipetting. Wonderful. Maybe you've done a more complex experiment because it's suddenly lifted the burden of all that planning and detail from you so that you can think about higher level things, which is absolutely what we want to enable. But also by doing this, we have created a map of exactly what's happened in that experiment. So when we get the data, then we can associate it with exactly how that data was produced, i.e. all the metadata which describes how that data point was produced. Right. And that's something which you essentially get for free because you've been working in the digital world throughout, right? You've been using digital tools at every step of the process.
Are you a biotechnology company looking to unlock the potential of your business data? CorrDyn can help. We're an enterprise data specialist that helps companies working in life sciences make smarter strategic decisions. From developing the right data strategy that starts with our data maturity assessment to building and delivering bespoke technical solutions, we are equipped to tackle the most complex data challenges. We have partnered with dozens of high-growth organizations, from manufacturers of custom oligonucleotides to molecular diagnostic companies, to achieve data competency. Whether you need to supplement existing technology teams with specialist expertise or launch a data program that lays the groundwork for future internal hires, you can partner with CorrDyn to unlock the potential of your business data today. Simply visit connect.corrDyn.com/biotech to learn more. Now, back to the show.
It seems like there's a lot of things that the Synthace platform is accomplishing as part of this sort of lab and research automation process, experimental design automation process. And it feels like at a high level, that data that you're capturing at the end, the metadata, along with the experimental results, is trying to improve the feedback loop that drives the research process, that drives organizational learning. When I think about dozens of researchers researching things in parallel, you want them building on each other's insights, not thinking along their own paths individually and chasing down wherever they're going. So is that part of the picture that you see emerging from the data?
Absolutely. The thing is what that picture that you're painting there is one that I think everyone aspires to, right? This beautiful record of exactly what went on, so that then if someone's doing something similar, they can hopefully even have the system just say, oh, hey, one of your colleagues did something similar earlier. Why don't you try this kind of thing? Which, to be clear, isn't in the Synthace platform yet. That's the kind of direction we'd want it to go, because as you're building up ever more of these structured and fully detailed data and metadata sets that describe all these experiments, and that's exactly the kind of foundation of data that we need in place as organizations to then make much more rapid progress. How is artificial intelligence going to understand about what makes a good experiment unless it's got access to those kind of data which describe a lot of experiments in full, right?
Yeah, that makes a lot of sense. Some of the benefits that I could imagine from this kind of platform would be increasing the speed to new discoveries, increasing the quantity of experiments that you can run, in parallel through the multifactorial design. Are you seeing customers of Synthace getting these sorts of benefits, and how have you seen it transform their
Yeah,
organizations?
it's been really quite cool because we make these tools and then you give them to scientists and they do like super cool things with them, and it never gets old seeing what people do with things. I think it's interesting what you say though, like in terms of running more experiments. It's actually we see people running fewer to get to a particular goal. Essentially, if you're like looking at one thing at a time and you're not parallelizing stuff in a multifactorial experiment, then you're doing iterative experiments and you actually have to do more experiments than if you can just parallelize everything. So it's a much more complex and much kind of higher throughput experiment that you're doing. And it's got like all of these multi dimensions to it, but it's basically answering the process a lot quicker than you would otherwise. And we found, particularly in kind of drug discovery, areas like assay development. So when you're first trying to work out the assay for high throughput screening or for screening for a new therapeutic, then that process of assay development can be really tough and it's highly complex and there's lots of factors involved. And what we're finding is that then when people are doing these kind of high dimensional experiments, then they're getting to the answer a lot quicker, because when you're working at that kind of scale of biology, because these biologists, they're used to working in 384 well plates or 1536 well plates. So that's 1,500 wells in an area like this big. For people who are listening and can't see me, I'm just holding up my hands in the shape of a 96 well plate. It's really a tiny scale. But what that means is they can do huge numbers of runs. And so if you can then use those runs to comprehensively cover a multi-dimensional landscape, then you can map out that landscape in exquisite detail and you can get the answer to what are the best conditions for my assay, or what are the best conditions for me to grow these stem cells, or what's the best way for my biology to work. You can get that answer even within a single experiment. And that, I was not expecting the platform to be able to be used for things of such power, because I wasn't thinking of doing 1,500 runs, right? Because I personally, like, my biology has not been at that kind of really small scale. So yeah, really cool scientists working with a platform, and frankly working with some really cool automation as well that can take things down to that kind of miniaturization, right?
So under the umbrella of a single experiment, you're able to do all of these micro experiments that provide information to each other in really intelligent ways, so that you can see the full picture of how what you're working with responds to the different treatments that you're providing in that controlled environment. And I hear you talking about that as both enabling discovery, but also enabling optimization of processes as well. Can you provide a little bit of insight into like how it works in those different contexts?
Yeah, it's mostly about when you have a system that you're looking to learn more about. There's some areas of biology where I couldn't see it applying quite so obviously, right? If you're looking for a new molecule that hits a particular target, then you're going to have to do some kind of medicinal chemistry or high throughput screening or whatever. And so this kind of high dimensional experimentation of the sort I'm talking about won't necessarily apply. But if you have something where we all the time, the things that we're using as biologists are the methods we're using are often highly complex, but we need those methods to be exceptionally effective because they are the foundation upon which the data is being built, right? So if we have poor methods, then we're not going to get good data. And so whenever there's a method that needs to be developed and understood and optimized, that's when these kind of methodologies really help out. I mentioned a couple there of assay development is one. For a biochemical assay, then there's lots of different components you might want to put into the liquid of that assay to make it optimized. And like, this is a brilliant way of working out the optimal mixture, right?
Sure.
Or similarly for growing cells, and then you go into like actually producing the drug substance, so bioprocessing, and this kind of thing's used all over the place. So again, optimization of processes it can be very powerful.
And another thing that I'm hearing is that this changes the relationship between a research and development org and their data. So it's asking them to think differently about their data. One of the insights that I appreciate is that there's a difference between data that's specifically designed to drive insight versus data that is just accumulating and then mined for insights. How does the platform facilitate that kind of relationship?
So I think that's a really key observation. And I think if we're just going to zoom out for a second, we think about like biology as a space and the kind of data that we need to understand biology. I think we've got a major disadvantage and a major advantage when we're talking about running experiments. A major disadvantage is that getting data from experiments is always going to be expensive. You compare it to, I don't know, getting data from social media to run some kind of AI to work out how to market something better or whatever. You can just harvest all that. But if you have to go in a lab to create every data point that you're going to make, that is going to be very expensive. The big advantage we have is we get to determine every single data point that we produce. So we can choose which data points we're going to produce in order to try and learn about a system. And so I think that then gives us very different opportunities for the kind of machine learning, the kind of like techniques we might use for exploring those data sets compared with when you have these really big, more amorphous sets of data, right? And we have those more amorphous sets of data in biology as well. Like very typically, the kind of data we come across is like these very big multi-omics data sets, right? I was talking with someone the other day, and they were talking about like the millions of different genomes they have sequenced, right? And, you know, that's a very useful type of data, but that's the one that's already quite well understood and that people understand the value of it and that kind of thing. And so I guess that's why I focus more on that. There's also the kind of this other much tighter, it's probably medium-sized data. It's not big data in that respect, and it's not small data, but it's somewhere in between. But it's highly directed because it's data which results from an exceptionally well-designed experiment, which is there to specifically drive insight. And I do think that is something a bit different.
So it's a different relationship to data. You're not trying to acquire as much data that exists in the world as possible. You're trying to curate a very targeted data set that is measuring the phenomena that you care about in order to accomplish the goal that you're working on, and designing the data effectively to accomplish that goal.
And it's because the data are expensive to make. And so I guess where the Synthace platform comes in then is we're trying to take away some of that expense, right? Hopefully like 90% of that expense, because a huge amount of it is like planning how to run the experiment, sitting there pipetting, trying to copy and paste all your data set together. There's just so much tedium which goes into your average biology experiment, which is frankly unnecessary. So can we make those data points cheaper? Can we make them more useful? Because often the data points you can produce by hand are going to be that you're limited by what you can feasibly do by hand. Right, you're limited by the complexity of what you can have in that 384 well plate, because if you've got stuff changing every single well, that's incredibly difficult to keep track of. And then someone comes up and taps you on the shoulder in the lab and says, "Oh, hey, did you order in those Falcon tubes?" And you're like, "Oh yeah, I did. They're on this shelf in the store cupboard." And then you go back to your plate and you're like, where did I get to? So there is just a limited complexity of stuff that you can do. So what we're trying to do is we're trying to just lift a lot of that burden from the scientists so they can do the experiments that really let them to get that insight into the biological systems they're working with.
So it's hard. I can imagine it's hard as a research scientist to zoom out from when you're having to do the manual, repetitive labor of petting to keep the strategic experimentation in mind of what you're trying to accomplish and how you're going to get there. I would imagine that you're freeing up a lot of mental capacity among research scientists to think bigger about what they're doing and the direction that they're taking their experimentation. My understanding of the platform is there's the design of the experiment and then an understanding, and then that gets outputted. I'm curious where experimental outcomes enter the picture for Synthace, because I'm imagining after the experiments are run, there's a process by which, you know, quality or purity or some outcome that you're trying to drive inside of the plate gets in there. Can you just give a little bit of insight into how you see those outcomes entering the picture?
Yeah, that's a really good question. So if we think about, okay, what enables that outcome, right, is essentially the data and the metadata. If you look at a lot of kind of XY plots of like of an experiment, then it's essentially metadata along the bottom in the form of the conditions that have been run or whatever, and data along the side. Y-axis data, metadata is the x-axis. So, you know, so long as you are collecting those things, then actually you can give scientists a window into the experiment that they've run really quite easily, and then they can see the outcomes. And sometimes that's easier than others. So we've got a partnership with Tecan, which is like the biggest liquid handling manufacturer in the world. And they've got this fantastic system for doing multiple purification runs simultaneously. It's called RoboColumns. So the way this runs is you have these columns on deck, and there's this liquid being injected into the top of them, and then it has a plate on the bottom that's catching the drops that come out of the bottom of the columns. So what the physical outcome is, you end up with a load of plates with clear colorless liquid in them, And those plates themselves are highly anonymous, just sat there on the desk, on the robot. And so you've got these anonymous plates, but like each one of those wells has material in it which pertain to a very specific part of that overall experiment, right? So in the physical world, these are all completely anonymous. And if you're not careful in the digital world, they're also going to be completely anonymous. So you have to have the metadata that understands, oh, this particular well pertains to this particular step that's eluting off this particular column, which had this sample applied to it. And then when you have that, actually, you could just concatenate those data points for that particular column really easily and just generate exactly the kind of output which a scientist would expect to see in order to then understand the outcome of their experiment, right? Mm-hmm. And then, so that's basic kind of data processing, data structuring. The next layer on from that is, okay, if I've run a really sophisticated experiment, then how do I understand the very sophisticated outputs of that experiment? So we also have capabilities in the platform which are specific to running high dimensional experimentation, specific to running design of experiments. So this multifactorial experimental design that I alluded to earlier. And there we actually go all the way from helping the scientists to generate that design in the first place, all the way through to building the models that come from the data out the other end. So that's about as far as we go. Most of the platform is just about generating those highly structured data and metadata sets, which can then be exported into other things for analysis. We see this as, we want to be the experiment engine, if you like, the thing that's producing the data. We don't have to be the place where all of that data is analyzed. We don't have to be the place where all that data is stored. This is way too big a problem for any one company to be dealing with. And it's inevitably going to be an ecosystem of all sorts of different tools that come together to solve the kind of overarching problem of data within any one of these big companies.
That makes a lot of sense. So if I'm understanding you correctly, the process, if they want to do multiple outcome assessments of a 384 well plate that comes out of this process, then they're exporting the sort of experimental parameters from Synthace and then marrying those up in their other systems with all the other tests they ran on the plates in order to understand sort of the relationships between the responses? Or are you typically seeing that the response that the user needs is in the liquid handler that you're interfacing with directly, or being passed back into Synthace for that kind of analysis?
It tends to be passed back in. So when you're talking about that specific experiment, anything that's really close to that experiment that's just been run, then that all resides within Synthace. So if they've done an analysis on a different bit of kit that we don't automatically take the data from, then they can just upload that and associate it with the experiment. And then it automatically, because again, it'll tend to be like 96 data points or 384 data points. We know what's in every single one of those wells because we put it there in the first place. So once again, you get this advantage, even if it's not something that's directly integrated into Synthace, it can be manually uploaded and associated. But where it becomes broader is, but what about the thousands of experiments that are being run across the whole organization? Are we going to then, and some of these experiments, I think, you don't necessarily need Synthace for them, in all honesty, right? If you're going to do some kind of genomic study or whatever, so it's mostly like sequencing or whatever, then the benefits of Synthace are probably going to be lower there. But we still want our clients to be able to take all the data from those experiments and the data from the experiments that are done on Synthace and put them into a much larger and broader data warehouse. That's what I'm referring to.
Yeah, that makes a lot of sense. So it's the point at which multiple experiments need to be married together with other things that are happening across the research organization that you expect the data to go outside of the Synthace system and for more customized, more bespoke, more organizational specific analytics to happen on top of those experiments.
Yes.
It seems like there's a certain amount of methodological evangelism that you have to do to convince people that the design of experiments, that multifactorial experiments versus one factor at a time is worth learning, is worth doing, is worth committing your organization to. Is that correct? And if so, what are the kinds of barriers that you face in trying to convince people of the value of that?
Yeah, it's all too correct. I think it just isn't used nearly commonplace enough that people just accept that it's a method, and everybody's been trained in a very different way to do science originally. I was trained in a very different way to do science originally. You can get frustrated at people like thinking, oh, why don't they just get it? But it took me 5 years. It took me 5 years from when I first heard about DOE to when I actually started using it. So I can't exactly say that I've got any kind of amazing prescience or anything like that. And so I do think that it's just a sort of thing which, yeah, it does require a bit of evangelism. It requires data. Scientists will always need data, and quite right too. And we have some absolutely brilliant data that's been presented by some of our customers at conferences over the years, like running 22 factor experiments to work out the best media for stem cells, or running single experiments to optimize an assay to a huge degree. There's all sorts of stuff that's been done in big pharma or small biotechs with our platform. So that really helps. But also, I think there is a bit of a wave of change. Like when we first started Synthace, then people had never heard of design of experiments at all, really, in my experience. And when you tried to explain it to them, they were like, yeah, that's your theory kind of thing. It's like they didn't have any sense that this is actually an exceptionally well established and highly effective branch of mathematics that's been around since the 1930s. But we ran a series of webinars last year on design of experiments because we realized that there was this kind of growing interest. And we just thought, okay, we'll put out this educational stuff. Barely mentioned Synthace at all. It just all be about DOE. And we got more registrants for the first webinar in that series than for the entirety of the previous year put together. And I think what that's showing is that there is a bit of a sea change. People are realizing that we need to be updating our ways of working, and whether that's by using more automation or digitizing things, or updating our experimental designs, or using more AI. I think more people are feeling the same frustration, right? And more people are impatient with this kind of attitude that actually things are fine, we just need to work harder, which seems to be like the almost unspoken thing in biology. Is like people really pride themselves on the fact that they've spent 12-hour days, 6 days of the week pipetting in the lab. And I think that is changing. And it's really exciting to see that change. But yeah, it's still going to take a lot more evangelism. And it's still going to take a lot more persuasion, but I enjoy it. So yeah, it's all good.
And at the same time, in the ether is all of the discussion about AI, machine learning, and how that's valuable for biology. And I'm just curious on your take on how AI factors into the mix of what's happening in research organizations that are the type of research organizations that would be using Synthace, could be using Synthace.
So first off, it's worth stating, I talk about DOE a lot, right? Because actually that is a great step change in the way people work. Automated DOE, even better, right? That's another step change all over from there. But actually, I see the future as AI. And Synthace as an engine is not a DOE engine. It's just an engine that allows you to run whatever experiment you need to run to really understand biological complexity. And that experiment can be designed by an AI, just like it can be designed, through a stats package. There's no fundamental conceptual difference there. But for AI to actually have the effect it could have, then it's going to need these highly structured, very well curated, fully contextualized, high quality data sets. And that's the way we can unlock the main potential for it. And I think it's going to be used all over the place in very different ways. So one of my frustrations a little bit is, actually, we've just been guilty of it in this conversation. It's just talking about AI as with this big blob, right? And it occurred to me the other day that talking about, oh, we need to use AI is almost like saying, oh, we need to use electricity. Right. It's only meaningful if you actually say, oh, we should use this particular form of artificial intelligence for this particular purpose. We should use large language models in order to help us to record our scientific intent a lot more effectively, and then to write up subsequent reports. We should use active learning to very effectively explore high dimensional biological landscapes, right? So like when we start talking in that way, then it becomes a lot more meaningful, and we understand the data we're going to need to make it run. We understand the kind of models that we need to be running to actually get benefit, and we can understand the kind of like change management, frankly, that we need to going to need to put in place to actually achieve these outcomes. It becomes a lot more like tractable, right? The moment that you're actually talking specifics. So I'm a keen proponent of that. Let's stop saying, hey, let's use electricity. Let's start saying, hey, this part of the room needs more light, so we're going to use electricity. We're going to put it through a filament, and that's going to give out light.
Yes. As I'm hearing you talk, the way that I hear machine learning or AI being targeted is being a part of the feedback loop that helps to understand the data that you have, determine what new data you need to gather, and then design the experiment to gather that data, and
Yeah.
then understand the data that you have and inserting it as part of the feedback loop between the research scientist
Right.
and the experimental data gathering that they're doing to
Yeah. And you
understand
just
what's
said something
happening.
super critical there, because you still kept the scientist in there, which is, I think that's really key, certainly for some biological systems. And again, I think we need to be talking about the specifics of the problem we're trying to solve. So there are some biological problems where, frankly, I say that a human might think they know what they're doing, but they're actually going to be actively detrimental. So I might offend a lot of people by saying this, but if you're doing protein engineering, then a protein is such a dynamic and incredibly complex thing that's working at the quantum scale, right? You are not going to be able to intuitively understand that system by yourself. Like, that's just not going to happen. I don't care, what you say. So humans should just get out of the way and let machine learning drive those kind of improvements. On the other hand, if you're developing a bioprocess, hell, I want that really wizened bioprocess guy who's been doing it for 30 years, because they've got a load of mental heuristics and models which are actually wholly valid. So depending on the system we're looking at, we should either be looking to how can we unleash AI in its full, or how could we have AI as the way that then we combine with human intelligence to actually get the most understanding from the experiments we're running. So I really like that you had the scientist in there, because I think for a lot of these systems, like, it makes sense to have that combination of artificial intelligence and human intelligence, and that's going to be where we get the most benefit.
That makes sense to me. And as I look at Synthace's take on DOE, it reminds me of it with the multifactorial studies and understanding like where the optimal regions are in that multi-dimensional space for a particular biological process to happen. It reminds me of parameter optimization methodologies in
Yeah.
machine learning. You've got this
Yep.
grid search approach where you're randomly trying different things, but then Synthace, as I understand it, is providing statistical tools for scientists to look at that and understand where are the areas of this multidimensional space where I could explore further and do more experiments. But there's this opportunity to look at the gradients, to look at the slope of these regions and understand, okay, these are areas of this space that it might be beneficial to push your experimental space out. Does that make sense?
Yeah. Yeah, absolutely. It's all about building that model which can help us to get insight, right? Whether that's artificial intelligence insight or human insight, ultimately all has to be human insight, right? But yeah, it's absolutely essential. And those kind of methods that you're talking about are highly analogous to exactly the kind of approaches that we advocate. But yeah, it's just with every iteration that we make, it's another experiment, and that has cost to it, and we're just trying to drive those experiments as efficiently and as effectively as possible.
As we bring this conversation to a close, just curious, what does the lab of the future look like to you? 5 years, 10 years down the road, what do you expect to see?
I've never been able to just conjure in my mind like an image of the lab of the future. I'm sure it's got some automation in there, and it's got digital tools and probably iPads and whatever, right? Maybe people going around with our headsets on. But I'm a lot less interested in technological solutions as like the methodologies that they're enabling. So a lot more interested in what the experiment of the future is, right? And how those hypotheses are generated, how the experimental design is generated, how it's executed. Actually, how it's executed is just the mechanics of it, really, isn't it? It's really about what will it look like to drive maximum insight and drive our ability to work with biology the most effectively. And for my part, it's going to be quite large scale, multi-dimensional experiments where you're measuring every possible outcome from that that's relevant to your system. Those very large, structured, high quality, deep data sets, then going in, like automatically piping into machine learning, which can then help to distill the huge amount of information that's there into the key aspects, which then humans can then interface with well and interrogate, before then having that sort of teamwork between man and machine, scientist and machine, to then work out what the next experiment is, right? So that's the thing that I have a clear vision of, and that it gets excited. The actual lab itself, I don't know.
Before we let you go, are there any books or papers that you've read recently that have stuck with you? Any material you would recommend to people as they think about this arena?
Wow, that's a fantastic question. To be honest, in my day-to-day work, I am dealing with so many different things that I don't have a specific thing I could talk about. I will recommend there's some great people writing in the space who are trying to unpick a lot of these kind of things. So they're not so much books, but there's some blogs and some people that are posting regularly on these things. So there's a chap called Nico McCarty who writes a kind of blog stroke newsletter called CorrDyn, and that's always got some fascinating insight. He's got some really deep thinking about the nature of science and the kind of biology we should be doing and the biology that's actually happening. There's someone on the data science side of things, a guy called Jesse Johnson. So he's got a fantastic series as well. And then there's a scientist at the Crick here in the UK, Erica Benedictus, and she actually has a lot of discussion and things about the nature of experimentation and how we should be driving this forward. And she's like super fascinated with cloud labs and how we can drive experimentation in the future. So I think those are the people and their output that really spring to mind when you ask that question.
Markus, thank you so much for joining us today. It's been a fascinating conversation and look forward to continuing it at some later date.
Yeah, it's been a real pleasure. Thanks a lot, Ross.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.